This video is learning targets 1 through 2 of Unit 4, Bacteria through Plants. It's kind of a broad unit and topics, but we're going to just break them down individually. So we're going to cover bacteria first. Before we do that, there are 6 different kingdoms that we're going to cover in this class, some of them more extensively than others. And so here they are: plants, animals, fungi, protista, eubacteria, and archaebacteria. You see the names there, they're Latin names that basically correspond with what they are. But the two that we're going to be talking about today are eubacteria and archaebacteria. Now quickly, a kingdom is just the broadest category of classification. This is where you see the broadest types inside of each kingdom, whereas in the animal kingdom you see everything from an ant to an elephant, a very broad category. And so archaebacteria and eubacteria are both classified as bacteria, and according to some, they are as different from one another as bacteria are from animals. So apparently there's a lot of diversity there, but we're just going to consider them very briefly in our study. Eubacteria and archaebacteria are both prokaryotic, whereas the other ones, plants, animals, fungi, and protists, are all eukaryotic, meaning they have a nucleus, they have complex organelles, and those sorts of things. So quickly, to talk about Archaea, or archaebacteria. Archaebacteria are the most primitive bacteria, meaning that they probably evolved first from the first organisms on the earth, and their cell wall composition is much different than the eubacteria. They also are able to inhabit lots of harsh environments. They're anaerobic typically. Here you see some scientists taking samples from a boiling spring, because bacteria, or these archaebacteria, can live in boiling water. They can live in salt mines, in acid pools. They can live near deep sea vents, like sulfur vents. And so they live in places that other things typically do not live. They're able to take up those sorts of environments and fit niches that other bacteria can't occupy. That's an archaebacteria. Eubacteria, on the other hand, more complex cell wall than the archaebacteria. There's more diversity in this group. They likely evolved from archaebacteria, or a common ancestor with archaebacteria. And so they're different, much different than archaebacteria, but they're going to have some of the same characteristics. One thing that's different is they have something called plasmids, and a plasmid is just a small piece of DNA. All bacteria have a chromosome, they just have one chromosome, but they can have multiple plasmids, and a plasmid just creates variation within the population. So whereas one type of bacteria, or one bacteria in a population, may have this plasmid that gives it immunity to antibiotics, for instance, and other bacteria may not have that plasmid, so that creates some diversity and variation in that population, which leads to adaptation and evolution ultimately. Plasmids are what we can use, humans can use, to engineer bacteria. We currently can make bacteria that will make hormones like insulin, human growth hormone, and bacteria can be even used to engineer things like plastics and other things. So in the field of bioengineering, plasmids are very important. Now, some organisms, just to introduce a new term, we've talked about photosynthesis, and photosynthesis is just simply organisms using the energy and light to create food. Well, not all organisms can see light. So under the sea, there's these deep vents that spew sulfur, and there are bacteria that corral around the edges of these sulfur vents, and they can actually use the hydrogen sulfide, or the H2S, which I have written here, to manufacture their own sugar. So they use this chemical to make sugar, and so that's why we call it chemosynthesis. Still making glucose, but just using a different energy source, using a chemical to do that, and thus creating different products. It actually makes sulfuric acid, which is highly corrosive. And so that brings us to this chart, which talks about all the different ways that bacteria can metabolize. Or what is metabolism? Well, it's just the sum total of all the reactions in your body, or all the reactions in an organism. This is how organisms break things down. This is how organisms build molecules. And we've talked about some of these, but we probably haven't given them these terms yet. I put a list of vocab up here at the top. Photo means light, chemical or chemo, chemical, auto, self, hetero, different, and then troph is feeding. And those basic terms can help us kind of derive what's going on down here on this chart. So let's look at this chart a little more closely. There are photoautotrophs. These are organisms that get their energy from the light to feed themselves. So they are able to get carbon from the outside and make it into their own food, and they use the light to do this. These are plants. This is the most simple kind of plants and things do this photo. They are photoautotrophs. Whereas a chemoautotroph would be an organism like we just talked about that do chemosynthesis instead of photosynthesis. They get energy from chemicals, and then they are able to fix carbon themselves. They're able to take carbon from the atmosphere to make glucose or carbon from their surroundings. Whereas a photoheterotroph is an organism that can use the sun to get energy or use light to get energy, but they need other things as a carbon source. They need to break down other materials in order to have a carbon source. And a photo or a chemoheterotroph uses chemicals to get energy and needs another source to get carbon. So chemoheterotroph, a good example of that would be like humans, animals. However, bacteria can be found in all four of these groups. Different kinds of bacteria can be found in all four of these groups. So bacteria are very diverse in their ability to take up different niches that are available. How do bacteria reproduce? Well, there's a process called binary fission, and it just literally means to split in two. And bacteria do not go through mitosis, the process that we've already covered, because bacteria are much more simple. They don't have the organelles, they don't have the cellular machinery to do mitosis. And so essentially what binary fission is just the replication of the DNA, making us another copy of the DNA, and then the cell wall and the cell membrane beginning to pinch in two, which is what we called cytokinesis. And so this is a much faster process than mitosis. And so this allows bacteria to reproduce much more quickly than eukaryotic organisms. And that's good for bacteria, but there's not a lot of variation because they don't have any kind of sexual reproduction. There's no mixing of the genes. There is one way that bacteria are able to do this, and this is called conjugation. And without going into all the details, it's not like sexual reproduction, like where you have male, female gametes that are combining to make a new organism. There's not a new organism made here. What's going on is two bacteria essentially join together and they exchange plasmid DNA. And so you can see this cell here on the left is making a copy of its plasmid and sends it to this other cell and allows these two cells to now be alike genetically. Whereas this cell on the right didn't have that gene, now it does. And so this creates variation in the population. Can be a very good thing for a bacterial population as far as evolution is concerned. So here are some different shapes of bacteria. There are the spherical shape or the coccus shaped bacteria, the rod shaped bacteria, bacillus, and the spirillum, which are spiral shaped bacteria. They can be arranged in different ways. The staphylo is like clumped up, so sometimes you've heard staphylococcus used together, and that's spherical bacteria clumped together. Or streptococcus, this is this like linear, this lined fashion here, but it's a coccus bacteria strung together, like strep means strung together. So those are the different shapes that are available in bacteria. And the bacterial cell wall can either be gram positive or gram negative. Now, what does this mean? Without going into a lot of detail, gram negative cell walls are much thinner than those of the gram positive cell walls. And notice that the gram negative cell wall, this purple line here, is also surrounded by two membranes, whereas the gram positive, the membrane is on the inside, much like we would think of a plant or something like that. Whereas the bacteria or the gram negative have a cell membrane on the outside and on the inside. This makes the gram negative cell much more difficult for things like antibiotics to treat. And so gram negative bacteria are typically much more dangerous than gram positive bacteria because they're harder to get rid of, they're harder to treat. Gram positive bacteria, fairly simple. Gram negative bacteria, much more complicated, much more dangerous. Now, what role do bacteria have in the ecosystem? Bacteria are decomposers in the ecosystem. What does this mean? They break material down to its most basic parts so that other organisms can then reabsorb those parts. If you think about all the things that you're made of cells, and those cells are made of all these different parts, phospholipids and DNA and amino acids. Well, the bacteria can tear all those pieces apart, can use what it needs, but then leave the leftovers for other organisms to absorb. And so, you've heard the whole circle of life idea where the lion eats the zebra, and then the lion becomes the grass and gets eaten by the zebra, and whatever else Mufasa said concerning that. Well, it's true in that the lion decomposes and then becomes food for the grass. And so bacteria are instrumental in doing that. Bacteria can also fix nitrogen. Now this, I wrote nitrogen fixation there. What is nitrogen fixation? This is just that bacteria are able to make nitrogen in the atmosphere, in the environment, useful by other organisms, most notably plants. Um, Nitrogen in the atmosphere we're not able to use, but bacteria can take it and make it into a usable form. Nitrogen in the soil that is there when an organism dies, it basically emits a lot of ammonia or a lot of ammonium, and that that's what that's why dead organism smells. Well, the bacteria can take that ammonium and make it into ammonia or a nitrate that is more useful by the other organisms like plants. Now, some plants, like this legume here, you can see all these little nodules. I had circles drawn around them. I'll draw it again. These different nodules here. These plants are basically create their own houses for bacteria. And so they have their own set of nitrogen fixing bacteria. And so these plants don't need to be fertilized as much as plants that aren't able to do this. Well, why do we have to fertilize in the first place? Because if you think about a field, a farmer will harvest that field and mow the field down and not allow those materials to be reabsorbed back into the environment. And so we have to put fertilizer, which is just essentially nitrogen, back into the ecosystem in order for new plants to grow. Because if we didn't do that, the soil will eventually be dead. Whereas like in a forest or like an open field or something, when organisms die, they just return to the soil and are constantly being recycled. So one way to do this in say a garden or something like that is to either take the dead material and compost it, which is put it in a big pile and allow the bacteria to break it back down into soil, or just allow those plants to die and be returned back to the soil naturally to keep the soil nice and rich in things like nitrogen.